Hi everyone, my name is Emily and my project is titled The Role of Interleukin-2 in Autoreactive T Cells. So I want to first begin by describing the research aims of my project. So I was really interested in understanding what the role of interleukin-2, also called IL-2, is in activated and memory T cells during the development of autoimmune disorders, specifically looking at a model of type 1 diabetes. I was also interested in looking at this question in vivo compared to in vitro conditions. So looking at this question in our mice versus in cell culture. And finally, I was interested in understanding if varying IL-2 environments would impact the regulatory T cell population at all. And to answer these questions, I used a novel conditional IL-2 knockout mouse model called the NOD-IL-2 flox mouse. And this model is really unique in that it allows us to understand the role of IL-2 in these activated and memory T cells, whereas previous models have only been able to look at the role of IL-2 in naive T cells, or T cells that kind of aren't fully functioning. So I want to provide a bit of background on this project, and I want to start with explaining kind of what T cells are. So T cells are a type of white blood cell that's important for the adaptive immune response, or the part of the immune system that provides kind of a strategic and specific immune response against certain pathogens. And there are two types of T cells. There are CD4 T cells, which either enhance or dampen the immune response by interacting with other immune cells, or there are CD8 T cells, which are known as killer T cells, and which I kind of have diagrammed over here. So the CD8 T cell is shown in purple, and the way that these T cells work is they release toxic granules into their target cell, which is shown in red here. So T cells can be involved in autoimmunity, and if you're unaware what autoimmunity is, it's when the immune system has a misdirected response, and the mechanism for recognizing self versus non-self in the body breaks down, and the immune system begins attacking its own cells and tissues. So in some cases of autoimmune disorders, it's the T cells that can become autoreactive. And there are a certain subset of CD4 T cells called regulatory T cells that protect against immune cells from becoming autoreactive, and therefore protecting against autoimmunity. So type 1 diabetes is in fact a T cell mediated autoimmune disorder. And what happens is that the autoreactive T cell recognizes and responds to the pancreatic beta cell by causing cell death and tissue damage. And these beta cells in the pancreas are what produces insulin. So that's why in type 1 diabetes patients, we have a loss of insulin production. And in order to study this disease in the lab, the preferred model is the non-obese diabetic mouse, or the NOD mouse. And really, this is the preferred model because these mice have very similar symptoms to humans. The way that their disease progresses is very similar to humans, and they also have similar genetic predispositions to the disease as well. So I've spent a bit kind of explaining the role of kind of what T cells are and their role in type 1 diabetes, but I want to also talk briefly about the role of IL-2 in T cells and also in autoimmunity in general. So IL-2 is vital for T cell proliferation, maintenance, and differentiation. So on the side over here, I have kind of a diagram of how kind of general IL-2 works for a T cell. So the T cell becomes activated and it begins producing IL-2 and producing a high affinity receptor. And the IL-2 binding to and signaling through this receptor induces T cell proliferation, producing a bunch of identical daughter cells during an active immune response. So it's kind of interesting when we started studying the role of IL-2 in autoimmunity that we noticed that both mouse models and humans with autoimmune disorders produce less IL-2, which is somewhat counterintuitive. We might expect the opposite. And further studies in mice show that if you blocked IL-2, this would result in rapid onset autoimmunity. However, this kind of paradigm was explained by the fact that IL-2 is critical for maintaining regulatory T cells, or those T cells that protect against autoimmunity, like I mentioned earlier. So there are two main kind of objectives to the methods aspect of this project that I want to get across to you. So the first is that I wanted to develop IL-2 deficient memory T cells using that novel conditional IL-2 deletion mouse model, the NOD-IL-2 flox mouse that I mentioned earlier. And then after I created these IL-2 deficient memory T cells, I wanted to inject these into mice without an adaptive immune system, meaning they have no B or T cells, and these are called the NOD-RAG mice, really to understand how these IL-2 deficient memory T cells function in the mice, and if they at all impact the T cell population in the mouse or impact the diabetes development of these mice. So in order to kind of create these IL-2 deficient T cells, I have to use what's called a retroviral transfection protocol. And the way that this works is that a retroviral vector, which contains the viral DNA, is combined with a packaging vector. And this is used to transfect phoenix cells, which these cells are a special cell line that produces a large amount of packaged retroviral particles into their media. And these packaged retroviral particles are what we need to induce this IL-2 deletion in our T cells. So these viral particles are able to infect our T cells and deliver a specific gene that induces this IL-2 deletion that we're interested in studying. And then there are several experimental methods that we can use to confirm this IL-2 deletion in our T cells. So once we have collected our T cells from our donor mice and done our retroviral transfection to produce these IL-2 deficient T cells, we're able to wash these cells and prepare them for injection into our recipient mouse, the NOD-RAG mouse. Once we do this, we monitor the diabetes development of these mice using either a blood glucose test or urine glucose strips. And then either once these mice are diabetic or after about 4 to 6 weeks, we euthanize them and harvest their spleens, their pancreatic lymph nodes, and their pancreas for further analysis. So I want to briefly and kind of generally talk about the results of these experiments for you. And unfortunately, kind of due to what's going on in the world right now, some experiments that we had planned had to be halted or just weren't able to start. However, we were able to optimize a lot of the protocols that were used in these experiments and kind of figure out the best way to, do the retroviral transfection protocol and what's the best way to kind of monitor these mice after we do this injection and how to also maintain these cells after we do the retroviral transfection. We were also able to show that these transfected T cells were in fact IL-2 deficient. And we were also able to show that we could maintain these IL-2 deficient T cells in culture before we injected them, which is was a really vital part to furthering this experiment because it allowed us to get better cell numbers before we injected our mice, our recipient mice. We were also able to show that once we injected these IL-2 deficient T cells, they could be maintained in the mouse, and that we could kind of recover these cells from the mouse and analyze them at the end of these experiments. We were also able to show that the retroviral transfection didn't change the functionality of these T cells, so they were able to act as normal T cells, and that this kind of transfection protocol didn't damage them in any way. And we were also able to show with very preliminary results that IL-2 deficient T cells led to changes in regulatory T cell expression and inflammatory cytokine production, both in culture and in our recipient mice. I want to end a bit talking about kind of the clinical significance of this project and why it's important. And it's really because IL-2 and its derivatives are currently being used in the clinic as immunotherapy to treat cancer and AIDS and autoimmune disorders. And specifically for type 1 diabetes, IL-2 derivatives are in clinical trials as treatment. However, use of IL-2 and its derivatives have been somewhat limited due to toxicity issues and the ability to selectively target certain T cell subsets, which might not be ideal for certain diseases. However, with more information about how IL-2 functions, we can hopefully figure out new ways to modify it to make safer and more effective drugs. Thank you. I want to thank Hans Dooms, my mentor, for his support on this project, and also Kilachand for supporting this project. And I think we can open it up to questions now.